Thanks everyone for coming, and thank you to the JGI for this invitation. So today I'm going to give you some highlights from work that we're doing in my lab related to this overall conceptual framework. So we try to understand plant microbial linkages as they pertain to the maintenance of plant diversity in ecosystems, as they pertain to ecosystem functions, and how they respond to and recover from anthropogenic changes, and we mostly focus on human land use. There are three aspects of land use that we've mostly been studying. One is urbanization. The second is agricultural land use, so trying to understand microbial composition and function within productive agricultural systems, and then also how these associations change over the course of recovery from historical land use. The first project I'm going to talk about relates to urbanization. And the reason why urbanization, I think, is an important area to study plant microbial assembly is because most of the global population resides in urban centers. And the 2014 UN report demonstrated that, and also found that global populations are only going to increase in their movement to urban centers. And so cities are becoming an increasingly really important part of the terrestrial landscape, and we know very little about their microbial ecology. And we know that urbanization alters a number of ecosystem processes and that microbes mediate a lot of the responses to these ecosystem changes. So I just started my lab at the University of Oregon in July, and for 8 years prior to that, I lived on this little island that you can see here. It's called Manhattan. And as you can see, the Manhattan in pre-colonial days, shown in the picture at the top, is very different than the Manhattan that we see today. So most of the vegetated, permeable surface has been replaced with asphalt and concrete, which is impermeable, and that not only modifies a lot of the ecosystem services, but also, of course, changes plant microbial associations. So we decided to take on this problem because there are a lot of practical reasons why we want to restore some of the ecosystem services in urban environments. One has to do with stormwater and wastewater management. So from the urban perspective, when you have a lot of people, there is a lot of wastewater. And a lot of the cities, especially older cities in the US, were not designed to accommodate the populations that we have in them today. So we have these systems that manage our wastewater. They're called combined sewer overflow systems. So that when we deposit our waste, it goes down a series of pipes that you can see a picture of what it actually looks like in the bottom there, some of the underground architecture of that. But then when there's a precipitation event, the stormwater actually combines with our wastewater, and when this reaches a certain threshold, there are outflow points that deposit that combined stormwater and wastewater into the surrounding waterways. And in New York City, we have about 6,000 miles of pipes underground that accomplish this task, and there are 422 outflow points around the city where when as little as 1/10 of an inch of precipitation falls, we can have an outflow event. So it's really a major problem for wastewater and stormwater management. It's only getting worse. And this is not just an issue in New York City. This is a diagram of some of the CSO areas that we have in cities around the country. Not sure if you can see the little dot near the coast of California, but that is San Francisco, where we are right now. So it's also an issue in this city. And it's also an issue in a lot of older European cities. So why am I talking about stormwater? Well, cities around the country and around the world, actually, who have these systems have been really pushing to implement changes into this gray infrastructure, and they're really starting to put a lot of resources and time and energy into designing green infrastructure, because that's a lot cheaper and a lot more effective than digging up 6,000 miles of pipes below places like Manhattan. So if we can actually use plant microbial associations to restore some of the ecosystem services, then we can effectively manage some of these problems in the urban environment. Now, from an ecological perspective, this offers a really unique opportunity to look at these engineered ecosystems and see if some of the basic ecological and evolutionary principles also apply to these ecosystems. Because in cities, when there have been studies of charismatic megafauna, they find that the cities are really homogenized biotically. And we don't know if that's the case for microbes. We're only starting to look at the microbial ecology of cities. So we wanted to know, do we just have the pigeons and the cockroaches of the microbial world in the cities? Are they all homogenized? Or are these really unique systems from which we can test ecological principles and then use that information to apply it to practical management strategies? So the research questions that we've been asking in the urban environment are: one, do microbial communities vary in predictable ways across green infrastructure type and location? So based on things like soil nutrient status or plant composition, can we actually make predictions about which microbes are going to be there? How are they assembled over time? Do we see successional patterns in GI microbes? How do they function in urban nutrient cycling processes? And finally, can we manage these assemblies to optimize GI function for the services that we want them to perform in the city? So we've been working in a series of experimental green roofs across New York City. The green roofs are indicated by the red dots on the map. And then we also have a number of parks we've been comparing the green roof communities to at ground level that you can see in the orange stars. So these green roofs were set up on the top of New York City recreation centers. The New York City Department of Recreation and Parks has been really helpful in helping us facilitate this experiment. We set this up in 2010. And for those of you who aren't familiar with green roofs, most of the time green roofs are planted with non-native succulent sedum plants. And that's just because landscape architects found that was a good plant. It's a CAM plant. It's really good at resisting drought conditions, at resisting a lot of insulation, which you find in green roof conditions. But from an ecological perspective, it may not be the best plant to put in these rooftop ecosystems. So what we did is we planted 2 native plant communities, Hempstead Plains and Rocky Summit, which are found in kind of rocky, mountainous areas that have similar environments to green roofs, and we thought might be good candidates to look at the native communities. We've also been surveying sedum roofs and mixed vegetation roofs across the city. So we've expanded out from just our experimental roofs that we actually set up. We've been looking at roofs that have already been established for a number of years. This project is being led by a master's student I still have left at Columbia, Jessica Hotch. And we've also been looking at other types of green infrastructure, not just green roofs, but also looking at tree pits, bioswales, which are specifically designed to capture stormwater, tree medians that are just in the middle of roadways, engineered tree pits, again, which are designed to capture stormwater, and parks, like the iconic Central Park that you see my students sampling in at the bottom right. We've been using Illumina sequencing to look at the bacterial and fungal communities in these systems. We've also been doing a little bit of RNA-seq on select functional genes and phospholipid fatty acid analysis, or PLFA. And PLFA, for those of you not familiar, what we do is we extract the lipids out of soil and we use gas chromatography. And what that does is it separates out the biomarkers that are microbial specific so we can get an estimate of living microbial biomass and also look at bacterial to fungal ratios. And the reason we want to do that in the system is because microbial biomass is often correlated with soil water holding capacity. And that's obviously one of the ecosystem services that's really prioritized in this urban environment. So I'm just going to show you a few highlights from our research. I don't have time to go really deep into all the projects, but one of the take-home messages is that urban green infrastructure microbes do indeed show habitat sorting. We are not finding biotic homogenization of these communities. So this is an NMDS plot of fungal communities that we sequenced from our experimental green roofs, from our ground level parks, and we included one of the parks we thought was going to be a ground level comparison, was the High Line. We published this in 2013, so you can go back and read more of the details or some other interesting findings there. But one thing I wanted to note is that the High Line falls out as kind of intermediate between the green roofs and the ground level parks. And I don't know if any of you have visited New York City and seen the High Line, but it's an elevated subway system that they converted into a park. So it actually kind of is intermediate between a green roof and a park, and we're seeing that reflected in the microbial community. So that was kind of fun to see. I do focus mostly on fungal ecology, but I know a lot of people study bacteria in here and just don't want to ignore the bacteria. We do sequence the bacteria. The bacteria are reflecting most of the patterns that we're seeing with the fungi. So as you can see, they also cluster out. The green roof bacterial communities are different from the park communities, and the High Line also separated out from those. We're also seeing habitat sorting and environmental filtering from our wider scale green infrastructure survey. So here we have the green roofs are separating out from the parks, from the tree pits, from the medians, and then we also have a forest control because we've done a little bit of work across an urban to rural gradient that I don't have time to talk about. But we do see lots of structuring, again, so we're not finding evidence of biotic homogenization. I had a former postdoc in my lab, Amin Gill, who did a project looking at specifically these engineered soils versus the non-engineered soils. And he was specifically looking at the compositional changes between them and the expression of functional genes that are involved in nutrient cycling. Also, specifically, he was interested in some of the genes involved in organic contaminant degradation, since that is a big issue in urban environments. And I should say that the engineered soils are made usually from proprietary medium mixes, like rooflight you will see, and they're also amended with compost, so it's not that they're devoid of microbes. They're just designed to absorb a lot of water so that when water flows into them, they expand, and there's also usually collection containers underneath the green infrastructure that allows time for the plants to transpire the water. And what we found was, yes, the microbial communities were very different between the engineered soils and the non-engineered soils. And also, interestingly, that the engineered microbial communities were much more diverse than the non-engineered communities. And this was the case for almost all of the functional genes at the level of DNA and also at the level of RNA. And he just published that paper. I'm not going to show you all the graphs. But one of the reasons we think that there's a diversity difference is because these engineered systems actually have inlets that facilitate the flow of the stormwater runoff into the soils there. So you are getting a really high diversity of resources for microbial taxa to target. And in ecology, there's this very basic principle of resource diversity and taxonomic diversity linkages. And we think we're finding that because you have more niche space for microbes to colonize. Whereas in the standard non-engineered soils, you just have the precipitation that's coming down. You have the stem flow, and you have just the water coming right into the system. You don't have a lot of stormwater that's running into those systems because they tend to run around it. So we think that that's a really interesting finding. And we also think that's good for urban management because it means that these bioswale and engineered communities are actually functioning well, they are cycling nutrients well, and they actually have a diverse and active pool of microbes that are performing some of the services that are desired for the city. We've also sampled our experimental green roofs from the time of planting in 2010 before we put any plants on them, every year in July at the same time. And we found a really interesting successional trend for these communities. So these are just the fungal data, but you can see that the cluster in 2010, that was when we sampled bare soil, bare media, and I call it soil, it's really growing media, but I'll say soil for the ease of speaking here. And that was before there were any plants. So that was, again, a proprietary mix amended with compost, so any of the microbes there were from the compost addition. We then have 2011 when the plants were added, and these plants were grown in a native nursery from seed with some soil from the environment, so there were microbes added there. Then all of that was combined, and we have not managed or irrigated or fertilized at all since then. So this has just been the natural succession of these communities over time. And you can see that they tend to stabilize. And 2011 or 2012 to 13 is when you see less change happening. And we were surprised because we sampled 2 months before Hurricane Sandy. We expected that to actually be quite a big perturbation to the system. It looks like it wasn't. So these communities are pretty stable even with storm events. And we're seeing that they are remaining pretty stable, although there is still temporal variation and some succession. Also, what was interesting is that the total fungal OTUs increased in the first 3 years, and then they seemed to plateau, and they haven't really changed. And this is true also for the Arbuscular mycorrhizal fungi that we've pulled out of our sequence data set. So we expected to see massive extinctions because these rooftop environments, they call them Mars-like environments. There's lots of sun, there's lots of UV, there's lots of drying of the media of the soils, but it seems like the microbes have been pretty resistant to that environment, and we have a somewhat stable community compositionally. So now looking at our larger green infrastructure survey, some of the data that we have in process is looking at microbial biomass differences across these different infrastructure types. And as expected, microbial biomass is highest in the forest. That's what we would hope. The parks and the green roofs are somewhat similar. And then we have really low microbial biomass in these ground level infrastructure types, so the tree pits and the medians. And we were racking our brains about why that is. We would think that tree roots would foster a lot of microbes. There would be really high microbial biomass in these communities. Same with medians. There's a lot of grass there. We didn't think that it should be that much different than a park or from a green roof. But then once we started observing these systems and doing a little more research, we realized that there is a different biotic influence at the ground level infrastructure types, and that is dogs. And we realized that there's a lot of visitation from dogs at these ground level infrastructure types, and that we actually did some research on dogs, and there are about 1/3 as many dogs in New York City as humans. That is a huge influx of external nitrogen into the system that's not happening in some of the other infrastructure types. We then did another project collaborating with an engineering PhD student at Columbia, Rob Elliott, and we looked at how microbial biomass and water infiltration rates were related, and they were correlated. And they also, both of those things, were much higher in tree pits that were fenced compared to tree pits that were not fenced. And the ones that are fenced prevent the dogs from visiting. So another indirect line of evidence that dog visitation is probably important. Well, we wanted to go a little bit deeper into this biotic influence. And so we actually set up a canine urine addition experiment. I do not recommend this in a non-ventilated system. And we had 2 of my undergraduate thesis students set up this project, and they added urine from 0% to 100%. They let this experiment run for a month. We used a plant that you frequently find in a lot of green infrastructure around the city. And what we found was that urine application decreased water holding capacity of the system, which is the figure I'm showing here. That's the average volume water runoff compared to the control. And you can see at about 2 and 3 weeks, the higher additions caused a lot more runoff. And we also found data I'm not showing you for the sake of time that we found decreased microbial biomass with higher concentrations of urine and also altered microbial composition. So it seems that, yes, urine is a big factor affecting the ecology of the ground level infrastructure systems, and we think this has important management implications for the city. While we can't fence off all the tree pits, we maybe need to do some irrigation to dilute the urine or have separate areas where the dogs are allowed to do their thing. Another project we have going on in the system that I wanted to highlight that's in process is an experiment again by my master's student, Jessica Hotch, where she set up a bunch of plants that are on green roofs. So we have sedum and then a couple of native plants that we find around green roofs in the city. And she sterilized the soil, planted the plants from seed, and is looking at the effects of different inoculum sources. So we're looking at inoculum from sedum roofs, the non-native plant, native green roofs, and park soils to see if there is an effect of inoculum source on the response and resilience of these communities to heat stress and drought tolerance. So that's something that's in progress, and hopefully I'll have something to report about that soon. And so ultimately what we're trying to do with all these various projects that a lot of them are still in the works is trying to understand if we can use the basic ecology of plant microbial assembly and function to not only ask ecological questions, but also to see if we can optimize the performance of these green infrastructure installations for urban management. And so it's been a really fun collaborative project. So that's all I'm going to say about our work on urbanization, because I also want to talk about some of the work we're doing in agricultural systems and also responses and recovery from historical land use. So most of the other work that we do in my lab takes place in a very different ecosystem in tropical rainforests. And tropical rainforests, I've been working on those since the beginning of my career, and I think they're really important ecosystems to study because they contain about 2/3 of all the plant and animal species on the planet. They cover only 7% of the Earth's terrestrial surface, but they have 50% of the biomass. They influence global climate, and land use is really pervasive in these systems. So I think it's a really critical ecosystem in which to investigate these questions. I've been working in a series of plots run by the Smithsonian Institute, the Center for Tropical Forest Science / ForGeo. They just expanded into the temperate and boreal forests, so they have plots outside of the tropics now. And these plots are fantastic resources for those of you who don't know about them because all of the trees in these plots that are greater than 1 centimeter DBH are identified, mapped, tagged, and recensused every 5 years. So it's a really great resource to ask questions of below ground ecology. And most of them are about 50 hectares in size, not all of them, but most of them. So I've been working in 4 of these plots, and I want to highlight some of the work that we've been doing in these, and I'm going to start with our work in Southeast Asia. So in Southeast Asia, one of the main concerns related to land use is the conversion of old growth forests to oil palm agriculture. And for those of you who don't yet know about the biodiversity crisis related to oil palm, palm oil is a commodity that is now the #1 most lucrative vegetable oil on the planet. And it's touted as a superfood. I would argue that it's not very super. It's also very diverse. It's in a lot of different products. You see it in most processed food. You see it in cosmetics and soaps. So very, very diverse product, and it brings a lot of money to some of these local economies. But as you can see, it's not grown very sustainably. What happens is that they clear cut the forest, they sell off the trees for timber because a lot of the trees in this area are highly priced for timber. And then they run these plantations, and they can run them for about 30 years without having to replace them. So they're very low maintenance, low input systems. And this is not just a problem in Southeast Asia, although 80% of our palm oil currently comes from Malaysia and Indonesia. It's a problem that is expanding across the tropics, and we're now seeing oil palm plantations popping up all over tropical ecosystems. And the reason that I originally got interested in Southeast Asia was not because of the oil palm crisis, but it was because there's a really interesting tree family down there that is a dominant component of the ecosystem, the dipterocarpaceae. This tree family is also very highly priced for timber, so it's heavily logged. But it's a really unique family in the tropics because it forms obligate ectomycorrhizal associations. And for those of you who don't know about mycorrhizal fungi, I just want to briefly highlight these. I figure most people know about them, but there are 2 main types of mycorrhizal associations in forests, the arbuscular mycorrhizal associations and the ectomycorrhizal associations. And the AM associations are found in almost all plants. They are very common from herbaceous plants to woody plants. Whereas the ectomycorrhizal association is much more restricted. Phylogenetically, it's been derived multiple times, but it's only found in about 3% of plants and mostly woody taxa. There are more fungal species that have been described from the ectomycorrhizal association. And what's interesting about the ecology of the tropics is that ectomycorrhizal associations are typically associated with dominant trees. So trees that end up outcompeting all of the non-ecto trees. And in other parts of the tropics, in the African tropics and in South America, you typically find 1 or a few species of ectomycorrhizal trees that become dominant. But the dipterocarp family is interesting because it's a whole family that's become dominant, and it's also had this radiation. So we have actually 600 plus species in Southeast Asia that are all coexisting. So it's a really interesting system to ask basic ecological and evolutionary questions, but also within the context of land use. So we've been looking at the effects of land use and also the ecology of the ectomycorrhizal association in this tree family in old growth forest, in forest that has been logged historically, regenerating forests. It was logging ceased about 50 years ago, and in active oil palm plantations. And we published some of our findings a few years ago now, but the take-home message that we found is that oil palm systems They do not have a reservoir of ectomycorrhizal inocula, so the soils cannot maintain ectomycorrhizal inocula. Ectomycorrhizal fungi do not associate with oil palms, of course. And so if the landscape is completely converted to an oil palm ocean, then we're not going to have a reservoir of genetic diversity for ectomycorrhizal fungi if we actually want to regenerate the forests, and that's really problematic. And then when we're comparing the forests that have been logged, the secondary regenerating forests, which are often considered degraded habitats, and they're actually prime targets for getting converted to oil palm agriculture, they do harbor an abundance of ectomycorrhizal fungi, but the composition is different. And so what we're interested in understanding is, why is the composition different? Is it because the abiotic environment of the land use change is shifting the fungal community, or is it because the tree community has changed and there's actually some level of host specificity between the dipterocarp trees and the ectomycorrhizal associates? And that's kind of the problem that we are trying to work out. So we don't actually know if there's host specificity in this system. A lot of the basic ecology has not been worked out in most of the species here. But there are two alternative hypotheses that we're working with. One is that trees and fungi segregate independently in response to similar abiotic gradients, and that's why you would see shifts across land use gradients. And alternatively, fungal shifts may be tracking the tree shifts across the land use gradients due to host specificity. So it's a very basic question, two basic alternative hypotheses that we're trying to work out. And so we've been trying to work these issues out in a plot in Borneo in Lambir Hills. And this is a really great plot because there is a very well-defined soil ecotone. So we can look at differences between soil variability and then also superimpose the biotic effect of host specificity on top. So we have these two major soil types that are clay and sand, and they are also subdivided. It's a very diverse plot. There are almost 1,200 tree species. About 70% of these trees actually have soil affinities. There are 87 dipterocarp species, so it's a really great place to ask these questions. And we know that microbial composition varies across the ecotone. We just don't know why. So there have been two studies on this plot looking at whether or not there's host specificity. One was a seed transplant experiment led by Sabrina Russo and analyzed by Kabir Pei. And they found that there was no evidence for host specificity. They transplanted seeds across clay and sand soils, left them there for 2.5 years, so it wasn't a very short experiment. And the ectomycorrhizal fungi segregated across soil type, but not across tree species. However, a former master's student in my lab that was advised by myself, and then Kabir was also highly involved, we had him do root tip sequencing of 3 canopy trees. One was a clay specialist, one was a sand specialist, one was a soil generalist. And while soil type was still the primary determinant of ectomycorrhizal composition, we did find some level of host specificity. So why is it that we're finding host specificity at the level of the canopy adult, but we're not finding anything at the seedling level, and we have no information about the size classes in between? So this is the area of research that we're really diving into now. And I have one idea that we have data from Puerto Rico to support this, that there may be actually what we're calling apparent host specificity driven by the traits of the trees. So one hypothesis is that there could be signaling between the fungi and the trees causing host specificity, but it could also be that the trees are creating chemical microsites that are resulting in unique habitats for different ectomycorrhizal fungi to colonize and thrive in. So that's what we're going to be testing in this system. And we're also going to be doing a much broader sampling of the ectomycorrhizal root tips on select dipterocarp trees and sequencing the genomes of the ectomycorrhizal fungi and doing some culture work to see if we can figure out whether or not we can predict their host affinity, their distribution, and their function. So that's kind of up and coming work in my lab. And the reason why we think there might be this tree effect creating this microbial signature is from work that we've done in Puerto Rico. And the plot in Puerto Rico has been a really good place to ask questions about land use because the southern half of this plot has low land use, and the northern half of the plot has high land use. And so we know that the land use in this plot alters microbial composition. We've already done that work, but we don't know why. And so what we did is we went in and targeted 5 tree species that occur on both sides of the plot that differ in their abundance and also differ in their traits. And we did a targeted sampling within what we would predict could be a potential microbial signature of that particular tree. And we sequenced individually these soil cores. And just briefly, I wanted to mention that we did find microbial signatures in both the soil, the mineral soil, 0 to 20 centimeters, and also the litter. So we separately analyzed the microbial communities in those two fractions. So these trees do have unique microbial signatures, and those signatures differ across land use types. So that's a really exciting finding. These data are from the fungi. We're right now processing the bacterial data. And I just wanted to highlight 2 of the trees because it's hard to put all 5 in one graphic space. So this is one of the dominant trees, Dactyroides, which has an affinity for the low land use area of the plot at the bottom. And then this is Cassiaria, which has an affinity for the high land use area of the plot at the top. And here you can see, I think it's just easier to visualize, the microbial signature, Dactyroides is in blue, Cassiaria is in green, and how that changes within a land use type and across the land use type. So we're trying now to figure out whether or not these microbial signatures relate to differential functions under these trees. And I have a grad student who was working a lot in my lab at Columbia, Andrew Quebeman, who's doing this work. He's actually looking at actual functions under these trees where we know the microbial signatures are different. We also just did some shotgun sequencing, and hopefully we'll have those data back soon to see if there are different abundances of functional genes related to nutrient cycling in these microbial signatures. And I think that this has a lot of really important implications for how we sample plots, and also how we potentially could scale up microbial function to the landscape. So if we can find that individual tree species have unique microbial signatures at the level of the tree species, and we can use information and new tools like hyperspectral data at the plot level where we can actually identify trees from large landscape surveys, then we could potentially use that information to better calibrate some of these global change models. So I've highlighted several projects that we have going in our lab related to plant microbial assembly across land use gradients. A lot of this work is ongoing as I just started at my new lab, so we're really excited about what we have going on, and hopefully we'll have some more exciting data to present to you in the future. And I also think it's been really great to be at this meeting and see some of these novel techniques and cutting-edge tool sets that we have in microbial ecology that can be used to address some of these basic ecological and evolutionary questions, but also to be able to apply those to practical management strategies in some of these degraded ecosystems. With that, I'd like to acknowledge co-authors and collaborators and my funding sources, and also thank you for the invitation here to speak with you today.